Recording in progress.
Hello, boys and girls. Good afternoon and welcome. It's been a while to our final episode of physiology. I had a lot of fun teaching you all this semester of all the things that I learned about the body, and I hope that anyone who's been watching and following along for the journey is inspired to learn more about the human body and what it takes to maintain a healthy lifestyle. And perhaps you picked up a few things that I learned and was able to enjoy learning. And without further ado, let's jump into some final review about the endocrine system. Excuse me. Endo meaning inside, crin meaning excretion. So this specific sphere has to do with the hormones that are released from different organs of the body, whether it be the hypothalamus or the pineal gland in the brain, or the liver, the pancreas, the digestive tract. We're going to be talking about all of those things. But first, let's talk a little bit about biomolecules and how they are digested in their own unique ways. Sugar is probably the most recognizable and easy to understand if you're familiar at all with glycolysis or metabolism, cellular respiration, that sort of thing. It's achieved by first breaking down these large polysaccharides, these chains of sugars, into absorbable monosaccharides such as glucose, galactose, or fructose. And this breaking down, this initial digestion is done by enzymes that are called brush border enzymes that can be found in the lumen of your digestive tract. So, for example, fructose, it is taken in from the lumen through a protein transporter called GLUT5, that's GLUT, whereas glucose and galactose go through the SGLT1 channel along with the sodium ions via a co-transporter that is Symport. And after all three of these different types of monosaccharides are brought into the enterocytes or the intestinal cells, they are then pushed or drawn into the blood through yet another glucose transporter protein called GLUT2, adding all three of them to the blood. So that's a little bit about sugar, and we'll talk about the effect that an increased blood glucose has downstream and what impact it has on the body. But I also want to discuss proteins and fats. So proteins as a whole, in certain cases, they may be phagocytized, meaning pseudopodia, these false arms kind of reach out from the enterocytes called the M cells, and they're just wholesale pulled into the cell and forced into a little vesicle bubble and sometimes brought straight into the blood, increasing BCOP, blood colloidal osmotic pressure. And that's important because it prevents certain conditions such as anemia, which is swelling in the feet, and it's a nutrition disorder. So it's through the the way that proteins are typically digested that we're familiar with. These proteins are digested, broken down through a chemical or a zymogen, meaning an inactive enzyme called trypsinogen, goes through yet another enzyme called enteropeptidase and is converted into active trypsin. And this active trypsin does a whole litany, a whole host of things. It uh, causes monopeptides, di and tripeptides to be brought in through the enterocytes. It essentially breaks down these larger proteins into monopeptides, meaning a single amino acid, not a chain, It goes through a symport channel into the enterocyte or perhaps M cell with sodium. So you get sodium and the monopeptide coming in to the cell at the same time. And there's also another type of channel that transports either di or tripeptides in antiport with protons. So as these specific types of very short peptide chains are brought in, these protons are coming out down the gradient. So you get this antiport action. And the symport channel of sodium and monopeptides that I described a moment ago, it's not only going from inside of the intestinal tract through the lumen and into the cell, it's also going from the cell and into the bloodstream. So really outside of a very special protein channel, you're not going to see anything outside of monopeptides being brought into the bloodstream. You don't want anything too big or charged coming into the blood so as to maintain order. Moving on to lipids, such as triacylglycerides, if you're familiar with the glycerol backbone and the three fatty acid tails coming off of it, that whole structure, if I can represent it like this, is digested by adding bile salts that come from the gallbladder. And what these do is they emulsify them or break them apart. They break them down into individual pieces and reorder them into micelles, just little spheres where the non-polar parts are kept on the inside. You get a bit of a hydrophobic effect going on. And colipase, an enzyme along with lipase, binds to these micelles to make individual fatty acid strands and monoacylglycerides. So you got the individual strands of this hydrophobic, very nonpolar substance, uh, be it probably a saturated fatty acid, uh, hydrocarbon chain, if you will. And then you also have the glyceride backbone that is still holding on to a single fatty acid. So fatty acid binding protein takes these, brings them through they're brought through the smooth endoplasmic reticulum, smooth ER, and then into the Golgi apparatus facilitated by this FABP protein. And they're rearranged yet again, if you can believe it, into tags triacylglycerides. It's just in the act of transport that they're broken down into their individual constituents and reordered together again. You can think of it as if you're with a group of friends and you're all locking arms, and then you come across a very skinny doorway. You just separate from your friends for a brief moment. It's facilitated by a leader. You all file through the narrow doorway, and then you all come back together on the other side. That's the best way I can describe it. And once you come through the other side, out of the Golgi apparatus and into the interstitium, the bubble that is now formed, this vesicle, if you will, is called a chylomicron. It's this little bubble of fat that has been emulsified, digested, and rearranged into the form of high density high density lipoproteins. I just had lunch. So through the interstitium, these chylomicrons are taken up through the lacteal structures and up into the lymph system, and then it's circulated all throughout the body, taken wherever it may be needed, storing fat for purposes that maybe serve your brain. Your brain, a large part of it is made out of fat. It's actually very important and easy way to store energy, very dense and accessible. Or maybe it's being stored somewhere less important, like in your arms, your extremities, or in your fupa. So what I want to talk a little bit now is to really hone in on the digestive tract after we've already started digesting through the saliva in our mouth, chomped it up mechanically with our teeth, swallowed it, gone through peristalsis through our esophagus, and gone through the cardiac sphincter and into the stomach finally. So I'm going to be talking about all of these various different cells, and I apologize for not having an illustration for you guys this week, but this is, if you can believe it, a very dense review. So you'll just have to bear with me and use your imagination this time. I trust that it's just as good or better than mine. So the first cells in the stomach, in no particular order because they're found really all around the stomach lining, is called the G cells. And what the G cells do is release gastrin, leading to a whole cascade of effects. In fact, every other type of stomach cell is activated by gastrin in one way or another. So these G cells, they release gastrin in response to a pH that is maybe getting a little too high, meaning your stomach's becoming a little too basic. We need to bring in some more acid so that we're able to fulfill our purpose as a stomach to break down this food with more acidity. And also there's the neurotransmitter acetylcholine that can trigger gastrin being released, perhaps through some neuronal activation, neuronal, however you pronounce it. And then there's also stomach distension. If your stomach's getting really full and you can't digest it fast enough, then your G cells are also going to be activated through that mechanism. Water break. So following the G cells, uh, we'll just kind of go down the list of different types of stomach cells. As I said, all of them are triggered by this gastrin that the G cell releases. Next, we have our D cells, which, when activated by gastrin, release a hormone called somatostatin. And what this really does is it slows digestion. This gives your stomach more time to break down the food before it's just passed through the rest of the tract so as to maximize the amount of nutrients that you take out of it. Next, we have the chief cells activated by the same mechanism. These release a zymogen called pepsinogen, which through another chain reaction becomes a chemical called pepsin, and then activates an enzyme called protease. Protease, as the name would suggest, is an enzyme that breaks down proteins, but it's actually not all that effective. In fact, it'll only digest about 8% of the proteins that come through, even when it's working at full capacity. So it's kind of on the weaker side, but nonetheless, it's an important part of the process. Next, we have the parietal cells. These are a real doozy. These are really important to understand because these are responsible for the majority of protein digestion. Proteins are extremely important for your body. Parietal cells are what create this molecule called intrinsic factor. What IF does is it allows your body to absorb vitamin B12. This is a vitamin that you cannot create on your own. You can only get it from meat, and intrinsic factor from your parietal cells is absolutely required for that process. Next, we have the ECL cells. To keep things simple, these release histamine, which then acts upon the parietal cells. So we get a little bit of paracrine action that propagates the cycle of digestion. So if there's ever a point where the parietal cells are slowing down, they're not functioning at full capacity, ECL cells activated by gastrin, maybe if parietal is not for whatever reason will release histamine and then kick it back into gear. And then finally, we have the mucus cells, which you can probably guess are uh, cells that release mucus. Pretty straightforward. These are exocrine cells, meaning they excrete into the rest of the stomach rather than endocrine, which releases into the bloodstream. The two endocrine cells are G and D cells, and the rest are either exocrine or paracrine. So once we have digested food in the stomach and it passes through the pyloric sphincter, it encounters the duodenum and the cells therein. Food at this point of digestion is called chyme. It's partly digested, but still not all the way there. We haven't been able to encounter bacteria in the small or large intestine, for example. But this chyme, this chewed up food, triggers cells in the duodenum, such as the S cells, again in no particular order. These are activated by interaction with um, hydrochloric acid, perhaps coming through the sphincter from the stomach. So an excess of that will cause secretin to be released. This is a hormone that activates what are called acinar cells in the pancreas, and these uh, activate enzymes and the release of bicarbonate to lower the acidity, to facilitate the storage or the export of carbon dioxide dissolved in the blood. And ultimately, this decrease in acidity will lead to less gastrin being produced. Next, we have the eye cells. These are known for being activated by fat or lipids passing through the duodenum. This turns on a hormone called cholecystokinin, and this does a good handful of things, but one of them is shrinking the pyloric sphincter, kind of trapping the food in the stomach, telling it you need to take a little more time to digest this food before passing it on, because we're detecting high fat levels that the body may not be able to digest, which could lead to conditions like steatorrhea. There is acinar activation by cholecystokinin, much like we talked about with the secretin coming from the S cells. There is satiation, so cholecystokinin will send a signal to your brain saying that you have enough food, you're full, you can stop eating. If you're eating fatty food, you may be familiar with that sensation. And then also there's bile acids that are released by the body as a result of high cholecystokinin levels, CCK. So if you can memory recall to a moment ago when we were talking about chylomicrons and HDLs, which go to VLDLs, LDLs, and LDLs. These triacylglycerides, they need to be emulsified, broken down somehow in order to be transported through the lumen and into the blood, or rather to the lymph system as a whole, and that is done by bile acids. Next, we have the L cells, which release a substance called GIP. And what this does is also shrinks the pyloric sphincter, slowing digestion effectively, and also activating the B cells in the pancreas, which release insulin, causing the body to soak up more sugar and store it for a later period. Next, we have the K cells, which act upon a substance called GLP-1. I believe these are actually the channels that I'm talking about, GLP-1 channels. Again, B cells, insulin are going to be stimulated, and this is also going to curb cravings So a little bit of satiation action going on. You may have heard of different drugs that act upon GLP-1 in the body, and these are effectively weight loss drugs because they cause you to eat less, not necessarily because they are exploding the nice cells of fat in your lymph system or something, but just telling your brain, hey, I'm not really hungry right now. A couple of honorable mentions: over-the-counter medications or maybe prescribed, such as cimetidine, which blocks histamine receptor action. You may remember that histamine is released by the ECL cells, which activate parietal cells, which uh, cause intrinsic factor to be released. So histamine is inflammatory. That's what causes swelling. Cimetidine may be used to reduce that by adding, well, actually, if you're blocking the histamine receptors, you want the histamine to increase in your blood. Excuse me. So you take cimetidine if you want more intrinsic factor, if you want to absorb more vitamin B12. That makes more sense. And then there's omeprazole. This is a proton pump inhibitor. This causes the channel that pumps protons out of the enterocytes and di and tripeptides in. So you are basically, if you have a ton of GERD, gastroesophageal reflux disease, and you have a ton of heartburn and want to stop it, you want to reduce or weaken the pH in your stomach that's causing this acidity to be thrown back up. You can take proton pump inhibitors, and um, the acidity will remain in your blood effectively. Okay, so that was a bit of a mouthful. That was our um, stomach, duodenum, pancreas, a little bit above the gallbladder. Now I want to talk a bit about hormones, specifically where they're derived from. There are three different classes we can talk about. There are protein-derived hormones, steroid-derived hormones, or singular amino acid-derived hormones. So protein-derived hormones are made in vesicles. They come in large numbers. Um, they are exositized from the cells, meaning they are just whole cell removed from them. They can circulate freely as they want, no strings attached through the circulatory system. They have a very short half-life. They are they serve their purpose within seconds, and they act upon members found or membrane receptors found on the membrane, such as G protein coupled receptors, um, tyrosine kinase receptors, another thing called JAK kinase receptors, which I'm not super familiar with, to be honest. Next, you have steroid-derived hormones. All of these come from cholesterol. Uh, they are made to order, meaning there's not a lot of them. Just as often as your body needs them, they'll just make them right away. They are not exositized, but they diffuse freely through the cells, meaning there's no sort of active transport going on. They simply phase through the membrane like the Terminator guy through the jail cell. The steroid-derived hormones, they circulate bound to proteins, so they're not as free as protein-derived hormones. And they have a long half-life. They act more on a function of hours, maybe even a day or so. And these act upon receptors found in the cytoplasm or in the nucleus of the cell. Um, these are your sterols, your estradiols, your testosterone, aldosterone, progesterone, estrogen, and the like. And the final and third class of hormones that I talked about or mentioned was the amino acid derived ones. These can either be derived from tyrosine or tryptophan, but tyrosine has two different routes it can take. Either it kind of goes more the protein or the steroid route. So if it's going the protein route, there's large stores of them. They're fat soluble. They have a long half-life. They act on um, these actually uniquely act on nuclear receptors rather than on the membrane and result in hormones like T3 or T4, which are thyroid hormones, which we will be talking about more in depth. The other type of tyrosine-derived hormones are stored in vesicles made to order, kind of like the steroid hormones. These are water soluble. These also have a very short half-life. They act on G protein coupled receptors, and these are ultimately catecholamines like epinephrine, norepinephrine. Those are the adrenaline hormones coming from the adrenal gland, obviously. Um, and then finally, we have the amino acid derived hormones. We have tryptophan. These are found in vesicle stores. These are less common than the thyroid hormones. They are water soluble, have a short half life, bound by G protein coupled receptors, and are released by the pineal gland. These are melatonin and serotonin, which act kind of in diametric opposites to each other. Okay, moving on, or about halfway through, I'm going to talk about the pituitary glands, the neurohypophysis and the adenohypophysis. So both of these are below the hypothalamus. The neurohypophysis is if we're looking at my brain this way, my adenohypophysis will be about right here. My neural hypophysis will be about right here. The difference is the adenohypophysis. This isn't the exact location, but it's made of these glandular tissues that uh, is the release site for many hormones, whereas the neural hypophysis is basically just a neuronal connection to the hypothalamus, not directly attached to it. But we'll talk about the neural hypophysis in the back first. This is the release site for hormones like antidiuretic hormone or vasopressin, as well as oxytocin, which is called the morality hormone, which is very interesting. And then in the adenohypophysis, up front here under the hypothalamus, there's several different types of hormones. We have growth hormone, regulating hormone. We have gonadotropin release hormone. We have thyroid stimulating hormone, CRH, and dopamine. CRH is corticotropin release hormone, I believe, and a whole host of downstream effects are facilitated by these. And I'm going to talk specifically more about GHRH, growth hormone regulatory hormone. So this is stimulated by certain conditions such as low blood glucose, REM sleep, meaning you're getting good sleep, and eustress, the healthy type of stress that you should be encountering day to day. So growth hormone regulatory hormone is released by healthy activities, needless to say. And as you can guess, growth hormone is a direct result of its activation, stimulates the liver to build up muscle and bone. Bone is also built up by things like prolactin and thyroid stimulating hormone and sex hormones. And another thing that growth hormone does besides building muscle and bone is also adipose tissue. This is a quote healthy type of fat, or rather, it breaks down unhealthy fat. So I mentioned TRH, thyroid regulatory hormone. This is acting in the hypothalamus up at the top of the brain. And then we have the TSH of the uh, adeno, or sorry, adenohypophysis. Yes, adenohypophysis. These thyrotroph cells that release thyroid stimulatory hormone. And then down all the way into the thyroid, which is actually about right here, uh, we release the T3 and T4 tyrosine derived hormones. And then there's a complete negative feedback loop. The more T3 and T4 we have in our system, the more it shuts down the thyrotrophs and the adenohypophysis and also the hypothalamus, creating thyroid regulatory hormone. So you don't want to convert all of these building blocks into T3 and T4 at the same time once you've reached a sufficient level or concentration in the body. You give yourself permission essentially to slow down the process, which is healthy. So if I were to receive some form of trauma in my, let's say in my thyroid, I get punched in the throat and I receive a lot of damage there, then we're going to see a sort of hypothalamic or hypothyroidism occurring. Despite my hypothalamus working fine, my adenohypophysis working fine, and their associated hormones that I just talked about, I might have really low T3 and T4 in my body. That is a hypothyroidism, and uh, it's easily diagnosable for those reasons. However, maybe if I have a tumor on my thyroid here, I might see a hyperthyroidism, meaning despite having very low um, T TRH and TSH being released, I still see super high levels of T3 and T4. And it's this negative feedback loop continues. The T3 and T4 just keeps getting generated despite the scarce resources. And the thyroid essentially ends up destroying itself from overworking. And I'm going to talk about a similar process that happens with the adrenal gland in a moment here. But first, I want to do a quick shout out to parathyroid hormone, which causes it's like these little dots that sit on top of your thyroid, and they encourage osteoclast action, which removes calcium from the bones and breaks them down to increase levels of calcium in the bloodstream. And calcitonin, I believe, regulates that. So your thyroid clear cells will release said calcitonin, and they will manage those low calcium levels. So moving on, we have uh, TSHR, thyroid stimulatory hormone receptor. This increases the activity of follicle cells. Follicle cells are found in the thyroid along with the clear cells I just talked about. But one such channel, or rather an enzyme that acts on a channel around the perimeter of these thyroid cells, these follicles, excuse me, um, is thyroid peroxidase. The enzyme acts on a channel called pendrin, which brings chloride into this follicular space, and it brings iodine out with a little free radical of its own. Hence that peroxide effect. Next, there is these channels called megalin channels. They facilitate the action of sort of an or phagocytosis. Essentially, it brings in these large thyroglobulin molecules, which are proteins riddled with iodine, and these are digested by proteases, as we talked about earlier. They turn these iodines, or they convert them into monoiodic tyrosine or diiodic tyrosine, and 90% of these will be converted into the T4 hormone, and then 10% will be converted into the T3 hormones, which are then received by nuclear receptors and are important for metabolism. These T3, or rather these 10% of monodiiodic tyrosines, could also be potentially converted into something called reverse T3 hormone, which is a, a mishap. It's a disease, and these are quickly eradicated by the body. But in any case, T3 and T4 are received by thyroid binding globulin protein and brought into the bloodstream so that they can be a part of the endocrine system. Okay, moving right along, I'm going to talk about the kidneys a little bit. Sitting on top of the kidney, we have the adrenal gland. If this is your kidney, this is the adrenal gland sitting on top of it. And the outside layers of this adrenal gland, kind of like an onion, are called the cortex. And the three parts of the cortex are called the zona glomerulosa, which, when activated, releases a mineralocorticoid, which is, let's see, this is the zona glomerulosa. So this releases aldosterone. And then next you have the zona fasciculata, which releases a glucocorticoid, such as cortisol. And then finally you have the zona reticularis, which releases another glucocorticoid, uh, such as DHEA, which is a precursor to the very important sex hormones. All three of these sit outside of the medulla. This is the heart of the adrenal gland. And the medulla produces adrenaline drugs, 80% epinephrine or pure adrenaline, 20% noradrenaline or norepinephrine. It's the same exact thing. So let's talk a little bit, kind of going back to the hypothalamus question. If my hypothalamus is stimulated, I'm going to have corticotropin release hormone in the anterior pituitary gland, or the adeno hypophysis. Sorry, it's backwards. Adeno hypophysis. And then ACTH is going to be released. And then in the adrenal gland, cortisol is going to be activated. So we have CRH, ACTH, cortisol that have this sequential order. Cortisol, kind of like we were talking about with T3 and T4, has a negative feedback loop. It'll shut down the actions of CRH and ACTH. And if we see hypocortisolism, that's evidence of trauma being sustained somewhere in the system. Or if we have hypercortisolism, that's indication that there may be a tumor somewhere. And those are that's not an exhaustive list of all the problems that could happen to your body, because there are a lot of things that could happen. That's a general idea. So cortisol is important for a couple of reasons. It's necessary for birth, for example. You can't deliver a child without a good concentration of cortisol. And also, cortisol, interestingly, is important for forming strong memories. You can see how this might be evolutionary advantageous. If you find yourself in a very stressful situation, you might want to remember that in the future so you don't paint yourself into that corner again. So, cortisol, those are a couple of arguably good, well, definitely good things, beneficial things that we could argue is responsible for. But it also increases blood glucose through questionable means. It acts upon the liver through a process called gluconeogenesis. It acts upon the muscle in your body indiscriminately causes proteolysis, breaking down of proteins. And it also breaks down uh, lipids in your limbs. So, I mean, having skinny arms might sound like a good thing on the surface, but at the end of the day, if it gets bad enough to the point where you are diagnosed with something like Cushing's disease, that fat doesn't just get vaporized and disappear. It'll reappear somewhere else, maybe in the back, and you'll get this buffalo hump. And you'll have very skinny arms, and you'll kind of get bloated. It's not very fun. So cortisol overall is only good in certain situations in the right doses. And it's also an immunosuppressant. So it's very easy to get sick when you're stressed. Moving on to the pancreas. This is the final part of the endocrine system. Then I'll talk a little bit about sex hormones and we can be done. The pancreas, as we talked a little bit about earlier, has these islets of Langerhans, including B cells, which release insulin, telling the body to store up glucose, uptake it from the blood, remove it from the bloodstream. And then there's also A cells, alpha cells. As opposed to the beta cells, they release glucagon, which tells your body to break down the stores of sugar, increasing blood glucose. So it's kind of hard to read my handwriting here. Insulin causes the liver to increase GLUT2 transporter action. So if you can imagine one cell in the liver, this is one specific um, uh, area in the body that will take up sugar through a very specific path. So insulin tells your liver to take up glucose from the bloodstream through a transporter called GLUT2. Uh, in your muscle, insulin will direct the cells to take it up through both GLUT2 transporters and GLUT4 transporters. So we see that the muscles, they have double, essentially, the access to sugar, which you can understand why. Um, and there's also an uptake of amino acids that goes on. So your muscles, as they consume more sugar, they also gain more access to amino acids, which makes it easier to build up protein in the long run. So exercise in the context of having sugar in your muscles is actually a good thing. You'd rather have sugar in your muscles rather than in your bloodstream. And that way protein is also built up in the muscle structure. And then finally in the adipose tissue, like we talked about in the limbs, this is only through GLUT4 transporters. So different from the liver, which is GLUT2. Again, that's GLUT. So now let's talk a little bit about diabetes. Diabetes insipidus um, means that there isn't necessarily, this is different from diabetes mellitus, meaning that your urine contains um, sugar in it. This is more about becoming dehydrated very easily. So diabetes insipidus is separate from type 1 or type 2 that you always hear about. This simply means that it's very hard for your body to hold on to that water. And that happens as a result of damage to your pancreas. But diabetes mellitus, on the other hand, is a little more insidious than insipidus. Type 1 is caused by the destruction of B cells, meaning that insulin isn't created at all. Um, and this can lead to ketoacidosis, your body digesting fat in order to find something to metabolize. Um, and then, whereas type 2 diabetes mellitus is that your body is producing a lot more insulin than you need. Your body is storing so much more sugar rather than uh, burning through it really fast, as opposed to type 1. Um, Kind of to be a little more thorough about type 1, if you don't have your cells in your body isn't producing insulin, there's no process telling your cells to store sugar. So it essentially just passes right through the body. And that's where the sugar in your urine comes from. Whereas type 2 is your body is creating a ton of insulin to the point where the B cells are getting burnt out, worn down. Maybe if you eat a ton of sugar, your body isn't able to keep up with it. And your B cells in your pancreas are becoming destroyed. Your body just needs to keep producing more and more insulin in order for that sugar you're digesting to be destroyed, or sorry, stored. That leads to problems such as AGEs, uh, advanced glycosylated end products, um, meaning that the collagen found in tissues all throughout your body uh, will become coated in sugar, for lack of a better term, and they will lose integrity. So this could be in your eyes, this could be in your nerves, this could be in your nephron, your kidneys, um, leading to blindness, kidney failure, um, loss of sensation in the extremities. So if you hear of people going blind from diabetes or losing a foot, that's the reason why. It's specifically type 2 in that instance. Very, very damaging disease. If you measure hemoglobin uh, levels of glycosylation in your blood, should not exceed 5% if you're a healthy individual. Okay, guys, we're down to our last part of the study here. We're going to be talking about the sex organs, the gonads, focus a little more on the female reproductive system, starting with the ovarian cycle. So if you can imagine the egg in the center, on the outside of that egg immediately you'll find a layer called the zona pellucida. Surrounding that you'll find the uh, granulosum, which is all of these little granular cells. And then surrounding those you'll find something called fecal cells. So the normal way of these egg cells and all of their various layers is that over the course of 180, 185 or so days, is that this these granulocytes, this granular layer around the zona pellucida will continue to expand larger and larger, slowly until these fluid vesicles form, these little bubbles within the granulocytes will take shape. And this is the point at which the recruitment phase, we call it, occurs. It's stimulated by the FSH, follicular stimulating hormone, and it will select only about 24 eggs out of maybe hundreds to move forward. And at the end of the process, there will only be one egg remaining, one very special egg, a champion, that continues to expand, take up all of the space with this super large granulosum. It just keeps expanding and expanding until finally it shears. It's ovulated. It remains this corpus luteum this giant mass of granulocytes and their thecal cells. They now have access to the bloodstream, being sheared off from the egg. They actually always had access to the bloodstream, but now the egg itself, now that its outer layer is removed, has access to the bloodstream itself. And after a woman, side note, menopause causes this corpus luteum to become a little shriveled up mass called the corpus albicans due to a lack of needed hormones to progress. So this ovulation leaves the newly sheared corpus luteum still in the ovary, whereas the egg kind of finds its way to the abdominal cavity, excuse me, near the uterus. So the corpus luteum, uh, this, okay, I'm trying to remember. It's called adrenos, ad, adenosteindion, something like that. The fecal cells, they bring in cholesterol from the blood, convert it to adenosteindion or stenodione. Forgive me for butchering that. But when they're attached to the granulocytes, they are able to move this adenosteindione directly into those granular cells, which then convert it into estrogen, which is uh, beforehand received by the luteinizing hormone receptor. And that estrogen produced by the granulocyte is brought directly into the bloodstream. And there's no gap between those sites. Whereas after the separation, after ovulation occurs, the fecal cells are also separated from the granulocytes. And since the granulocytes now have clear access to the bloodstream, that cholesterol can come in and create progesterone also, which is released into the bloodstream. So as this egg develops, the more the female body releases progesterone in anticipation of a pregnancy of a fertilized egg. So moving forward, the egg is in the abdominal cavity. Estrogen and progesterone increases. Estrogen is known as E2, progesterone is known as P4. And let's say that the egg is fertilized, it'll begin to release a hormone called uh, human, uh, called HCG, H standing for human. And this is close enough to luteinizing hormone to actually continue the process of releasing progesterone and estrogen into the female's body um, to activate the receptor that I mentioned. But how does this fertilization happen? The sperm has a head, an acrosome with this acidic substance that can eat through the zona pellucida of the egg. Um, it has a nucleus on the inside, of course. It has a centrosome, uh, which interestingly does not come from the egg. Centrosomes that are responsible for continued replication of the fertilized egg cell come from the sperm. There's a mitochondrion spiral connected to the flagellum, and it's a pretty simple structure. The the uh, the sugar that the sperm metabolizes comes from the seminal vesicle, and the mucus that accompanies the sperm in the form of semen comes from the bulbourethral gland. The testes follow the vas deferens up up and around towards the prostate and then out through the urethra. And in the testes, there are very important cells that bears mentioning called the Sertoli cells that release androgen binding protein that essentially binds to testosterone and keeps it local to the testes. It uh, keeps it concentrated and uh, prevents it from wandering throughout the body a little too far. There's aromatase, also in the Sertoli cells that convert testosterone into estrogen by aromatizing them. And then there are also inhibins, which complete a negative feedback loop. So we have our gonadotropin releasing hormone, which releases, so that's in our adeno hypophysis uh, in the under the hypothalamus. Males also have FSH and LH, which then give rise to the action of Sertoli cells and something called Leydig cells. Leydig cells produce testosterone and DHT, which is the more powerful form of testosterone. The testosterone prevents luteinizing hormone and GnRH from functioning, whereas the Sertoli cells, the inhibins that they produce, they only inhibit the FSH. So that was our exam review. Thank you for listening along. I had a very fun semester learning and sharing with you all, and I look forward to watching these videos back, and I'm very thankful for you all being patient and learning. Hope you have a nice life. See you later.